I'll give you plenty of time to ask questions about hemoglobin. So the new stuff that we cover on hemoglobin today is not on the exam, but we're kind of just wrapping up the story. Okay, and so hopefully it will solidify what we talked about last week. So this is just a reminder. So what we've already talked about is the structure function. So what do you mean? What do you think I mean by the structure function relationship? So how is the structure from myoglobin similar to and different from the structure of hemoglobin? Okay, so myoglobin is a monomer that exhibits hyperbolic binding of oxygen. Hemoglobin is a tetramer that exhibits sigmoidal binding of oxygen. We also call that cooperative. Okay? How does the structure of hemoglobin, being different from the structure of myoglobin, contribute to that functionality? Okay, so the affinity changes. How does the affinity change have anything to do with structure? So I guess I could also say, if we say there's a conformational change in hemoglobin when oxygen binds, is there also a conformational change in myoglobin when oxygen binds? Yeah, there is. Okay, so when the oxygen binds, you still have that alpha helix moving a little bit to put the histidine closer to the oxygen, or the iron, which is in the plane of the porphyrin ring, right? So you still have a conformational change. How is that, though, not cooperativity?
It's only one subunit.
Okay, so there's no other subunits around it to sense that conformational change. Okay, so myoglobin still undergoes a conformational change when oxygen binds, but nobody else knows about it. Okay, so all the other molecules of myoglobin aren't close enough to be influenced by that molecule of myoglobin. So in hemoglobin, when oxygen binds to one of the subunits, conformational change, just like in myoglobin, but now it's in a crowded situation. So the other subunits are around it. The other subunits sense that conformational change, and it makes it easier for them to almost, and here's where the debate, and it's still not entirely known, so if subunit 1 binds a molecule of oxygen, does that pre-shift subunit 2 to the R state before it binds oxygen? Does it expect an oxygen to come so it changes conformation even before it sees the oxygen? Not necessarily. It just makes that transition easier. So that's the cooperativity. Okay, so parts of myoglobin are similar to hemoglobin and parts are different. Okay, so that's the structure-function relationship. The function in terms of under what conditions does hemoglobin bind oxygen, that functionality is related to its structure. Okay, so what would you say is the function of hemoglobin? To carry and deliver. Okay, so myoglobin, the function is to bind oxygen. Hemoglobin, it's not designed only to bind. It's to bind and deliver. Okay, so it's the transport and delivery more than simply binding. Okay, so they have different functions that are consequences of their structures. So how does tertiary and quaternary structure affect that? So again, the quaternary structure of hemoglobin is what allows the cooperativity. You don't have cooperativity if you only have one polypeptide. Talked about conformational change. So regulation by small molecules is kind of where we are now. And pH. Okay, so sit tight with me for a minute while we review some buffer conditions. Okay, so if you didn't know it already, the buffer in your body that maintains your blood pH is a bicarbonate buffer system. Okay, so this H2CO3 is carbonic acid. So carbonic, gotta tell my pen it's supposed to be a pen. So H2CO3 is carbonic acid. HCO3 minus, what do we call that? Bicarbonate anion. And that's still acidic. It can still go and lose that second proton, and then we call it carbonate. Okay, but really the only pKa that we care about is that first proton. Okay, so the pKa of that first proton of carbonic acid is 6.1, so that's in the physiological range, and so that's the relevant one. So if I told you that in your body you have 20 times more bicarbonate anion, so the conjugate base, carbonic acid, than you do of carbonic acid, you could predict for me, calculate what the pH of your blood plasma is, right? So the pH equals what? Okay, so 6.1 plus log of what? 20. So 20 over 1, right? And so what's the answer then? What's 6.1 plus log of 20? It darn well better be 7.4. Okay, so your blood plasma pH, we've already talked about physiological pH. It's not exactly neutral at 7, it's 7.4. So it's because this is the buffer system and because we have an excess of the conjugate base. Why do you think it makes sense that we have an excess of conjugate base compared to acid? Physiologically, why do we need...
For to balance the level.
Okay, so what are we... You want to add to that answer? So what are we most likely to produce in metabolism? Acids. Yep. Okay. So when your muscle works, especially under anaerobic conditions, what do you produce? Lactic acid. Okay. Oh, it's right there. So lactic acid, then as a result of metabolism in your muscles, is going to, so it would lower the pH, right, because it's an acid, except now we have this bicarbonate system. Okay. So we have the bicarbonate conjugate base, so now we can kind of exchange those protons. So the protons that were coming from the lactic acid now can be taken up by the bicarbonate conjugate base because we have so much of it around. So are there any metabolic reactions that produce base? Not so much. No. They're almost all acid producing. Okay, so if we then make, so where does the lactate go eventually? So we'll get to this, I promise, in metabolism. So if your muscles make lactate, they go back in the bloodstream. They go in the bloodstream to the liver. The liver converts it back to pyruvate to continue the citric acid. So we'll talk about that later. That part's not what you should know. But what you should know is we have this bicarbonate buffer system to deal with the metabolic byproducts that are acidic, really. Okay, so then we make this carbonic acid. So we kind of shift the equilibrium to make acid. And we have an enzyme called carbonic anhydrase. Carbonic anhydrase then will interconvert carbonic acid and carbon dioxide and water. Okay, so it turns out that you have carbonic anhydrase both in your muscles and in your blood, kind of near your lungs. Okay, so carbonic anhydrase goes both directions. When we start talking about enzymes today, you'll see enzymes are typically reversible. So if we have carbonic acid that's being produced in our muscles by kind of mopping up that lactate, that lactic acid, now we're going to make carbon dioxide and water. Water's no problem. Do we want a lot of carbon dioxide bubbles in our muscles? No, that's not good. Okay, so what can happen to the carbon dioxide? You're going to see it's going to hitch a ride on hemoglobin. Okay, so we'll see that in a second, but I first want to come back to the bicarbonate buffer system. Does everybody kind of get why this is a big deal? Okay, so one of the first things you do if you're in a hospital and kind of checking somebody out in a crisis situation is their blood pH off. Okay, so that's often an indicator that something wacky is going on, because if you have a pH imbalance, you better figure out what it is. Okay, somebody who's acidic or ketotic, so often that indicates diabetes, so a diabetic crisis. What's that? Ketoacidosis is like, woo, we got molecules there that don't belong, and we better deal with it. And so often you would, you treat with kind of a bicarbonate situation. Okay, so that's the bicarbonate buffer system. Questions about that? So the details about carbonic anhydrase, you don't need to know that. It's mostly just the bicarbonate buffer system that's important at the moment. So I'm going to tell you about the Bohr effect. So you may have heard about that, I don't know, cell biology or elsewhere. The Bohr effect just says under more acidic conditions, hemoglobin changes its functionality a little bit. Okay, so if you were to plot the oxygen binding curves, the sigmoidal binding curves for oxygen to hemoglobin at different pHs, hemoglobin will unload its oxygen differently depending on the pH of the solution. So that's called the Bohr effect. Okay, so if we say our tissue has a PO2 right around there, so if we have a slightly acidic situation, does that favor the T state or the R state of hemoglobin? T state. Okay, so if this is normal for pH 7.4, if we have a lower pH, we have lower oxygen bound, more oxygen delivered. Okay, how is that a physiological advantage?
I guess maybe going to increase metabolism, that release more oxygen.
Right. Okay, so if your muscles are working hard, lowering the pH locally, that probably indicates you need oxygen there. Okay, so if you're sprinting, you aren't getting enough oxygen to your tissues, your pH of your blood decreases in your tissues, that's kind of like a signal that, oh, we better get more oxygen to the muscle tissue so that we can continue respiration. If we have a slightly higher pH, again, that doesn't happen too often, but now we're going to hang on to the oxygen more. We really don't need oxygen in the tissues quite as much. Okay, so this oxygen binding curve shifts a little bit depending on the pH. So T state favored, R state favored. Okay? So at lower pH, and typically that's higher carbon dioxide concentration, they kind of go hand in hand, because if you're metabolizing fuel in your tissues, you're lowering the pH and producing CO2, right? Because of this. Okay, so we're lowering the pH and making CO2, both of which we need to get rid of. We need to get rid of the protons, we need to get rid of the CO2. Okay, so what happens to the protons? So a surplus of protons, they will protonate one of the side chains in hemoglobin that favors the T state. So again, here's where structure and function come together and where chemistry comes together. Okay, so you have a histidine side chain around a pK of 6. So if we start to lower the pH, there's more protons around. Now the histidine is more likely to be charged. And we have a positively charged side chain. It can make a salt bridge with a negatively charged side chain. That's going to be a conformational shift to the T state. So low pH favors the T state, which favors oxygen delivery. And that's the Bohr effect. Questions about pH and hemoglobin? Okay, so now what happens to the CO2? So we already kind of dealt with the protons, right? So they're going to jump on to hemoglobin, favor the T state. Now the hemoglobin is going to go back to the lungs to pick up more oxygen. But what about the carbon dioxide? We don't want a whole lot of carbon dioxide bubbling around in our tissue, right? So it turns out, it's kind of wacky, the carbon dioxide also hitches a ride on hemoglobin. Okay? So if this is the N-terminus of one of the 4 polypeptides in hemoglobin, okay, so this is, we're going to call that C number 1. So that's the first amino acid in one of the polypeptides. We have the CO2 there. Now look, we're going to actually make a covalent bond on the amino terminus, and this is called a carbamino terminus or a carbamate adduct. Okay, so carb is the CO2 part, and then the amide, carbamate, is, so this is a carbamate, basically. Okay, so now, so we're going to release a little bit of proton. That can go off and protonate that histidine side chain. That's all good. And now, where's the CO2 going to go? So the CO2 is on the hemoglobin. Where's the hemoglobin on its way to? The lungs. And what happens when you get to the lungs? Okay, so partial pressure of oxygen increases. We favor the R state. Everybody changes conformation. And now it goes the other way. So now we're going to release the carbon dioxide. Okay, the histidine residues will be deprotonated to favor the R state. And now the fact that you have carbon dioxide in your lungs, that's a good thing. You're going to breathe it out. Okay, so the carbon dioxide that you breathe out really came from metabolism in your tissues, and you're exhaling it. All through hemoglobin. Everybody okay on that?
So it doesn't interfere with the oxygen from?
No, so it's a different place. Okay, so here's a great example of allostery, right? So it's another place. So often allosteric effectors are not covalent. This one happens to be covalent. But it's binding somewhere else. It's not getting in the way of the oxygen. When it gets to the lungs, now it just reverses. It comes off and gets exhaled out, and now the hemoglobin can pick up more oxygen. But that's not because they're competing for the same site. Okay? So we already talked about BPG, right? So BPG is this funny little molecule, 2,3-bisphosphoglycerate. Don't learn the structure now. You know it by the end of the semester when we talk about metabolism. And it is, we already said, a heterotropic modulator. So it's a heterotropic negative modulator of oxygen binding. So it's going to shift or stabilize the T state because it sits right in that core of the hemoglobin. And only in the T state does hemoglobin have that space there, so it kind of locks it into place. Okay, so I asked you why is it helpful that your body makes more BPG at high altitudes? Okay, so if the oxygen partial pressure at high altitudes is lower, even if you're getting as much oxygen to your hemoglobin as you can, there's just not as much. So it's kind of like the saturation curve. You know, if this would be normal, oops, that's not so flat. So if that was going to be normal hemoglobin binding, now at high altitude we just can't, you know, the PO2 of oxygen isn't as high, so we can't fully saturate our hemoglobin. So if we make more BPG, how does that help us? Does it help us bind more? No, there's not more to bind, really. What does it help with? It helps deliver more. Okay, so if the BPG shifts to the T state, now we're kind of like wringing all the oxygen out of the hemoglobin that's there. So we can't bind as much, there's not as much in the atmosphere to bind, but what does bind gets delivered more efficiently. You're squeezing all the last little bit of oxygen out. Okay, so you can kind of train yourself to deliver oxygen more efficiently.
And is this for like marathon runners and things too?
Yeah, so lots of athletes train in Colorado, you know, at high altitudes to increase. So you're forcing your body to work at low oxygen, and so your body says, well, I know how to compensate for that. I'll make more BPG, and then the effect lasts, I don't know, a couple of weeks after you leave the high altitude, and so you still have residual BPG. Then if you compete at sea level, you know, you can just more efficiently transfer back and forth.
This mechanic is like blood doping?
So apparently, so this is physiological. This is natural and is not doping. So this is not illegal. Somebody told me, and I haven't looked into this, I don't know much about doping apparently, that you can actually consume BPG or some precursor to BPG without doing the high altitude training. So there must be a test for what's your blood BPG level, and like, you know, this is if you train at sea level, this is if you train at high altitude, and then there's like this if you dope. And so I guess they must be able to detect whether it's just high altitude training or whether it's artificial and illegal. I kind of think that at some point it's not going to be a huge advantage to dope with BPG because, and I don't even know, like if you just ate this, I don't think it would stay in that form. Like there must be some precursor that makes this. I'm not sure what it is. But, you know, it may be one of the things that they can test for. I thought I knew a lot about doping, but having never done it, perhaps not. So, and again, this is just, you know, a space-filling model of the BPG binding site. So T state, R state, you can see there's just no room for it in the R state. Okay. Now, what if we have carbon monoxide instead of carbon dioxide? Okay. Bad molecule, right? So, in what way is carbon monoxide more similar to oxygen than to CO2? Well, so it looks more like oxygen, right? And so where is it going to bind? So it's going to bind where oxygen is supposed to bind. Okay, so it's going to be a competitor for oxygen. And so if we now have some CO present, it's both taking the place of oxygen, so you're preventing the oxygen from binding, and it turns out it binds more tightly. So it makes it harder to deliver what oxygen is there. So you don't need 100% carbon monoxide to die. You just need a little bit of carbon monoxide. So that's why it's so dangerous. So, you know, a little bit of carbon monoxide that you breathe in, it doesn't have to see all of your hemoglobin. Not all of your hemoglobin has to be saturated with it, but you can't bind as much oxygen, and the oxygen that you do have bound isn't delivered as efficiently. Okay, so do you see where where the curve kind of shifts over into here. Okay, so if you have carbon monoxide, you're holding on to the oxygen that you do have, even at low oxygen concentration. Okay, so if you're anemic, which means you don't have as much iron as you should, and so you can't bind as much oxygen, so your oxygen binding is less, even if there's plenty around, but you can still deliver it better than if you're poisoned with carbon monoxide. Okay? Questions? Yes?
Yeah, so what about carbon monoxide that allows it to bind irreversibly to hemoglobin? And just doesn't have-
Well, so I don't know that it's irreversible, it's just tighter.
Okay.
Okay.
So it could be removed in something like it could be released or donated somewhere, but how?
You have to get to the hydroxy.
Right, so if somebody is carbon monoxide poisoned, you have to get them somewhere, so how much oxygen is there in the air?
20%.
So it's only about 20%. So if they're in an environment that's more than 20%, so hyperbaric chamber, right? Now you're kind of forcing the hemoglobin to see even more oxygen than just air. So getting them to air first is important. But then putting them someplace where you're kind of forcing the carbon monoxide off just by flushing so much oxygen over their hemoglobin, that will help to bump off the carbon monoxide. What else was I going to say about that? Oh, so how would you classify carbon monoxide as a modulator? So it's definitely a negative modulator because we're not binding and delivering oxygen as well. But the weird thing is it's binding to the same site. So it's almost like, so I don't want to confuse you, but it's almost like it's forcing the hemoglobin to look more like the R state. So the hemoglobin thinks it's binding oxygen because it's binding in the same place. So you're binding an inhibitor but trapping it in the R state. So it's not releasing it as easily. So it's definitely heterotropic because it's not what it's supposed to be binding. It's definitely inhibiting because you're not binding oxygen, but you're also shifting it to the R state structurally. So I guess you could say carbon monoxide uncouples the structure-function relationship that's supposed to be there. So it's staying as an R state, even though you really would rather shift it to T to deliver. So if that confused you, ignore what I just said. Any other questions about hemoglobin? Okay, so the last kind of structure-function connection we want to talk about is sequence to structure to function. Okay, so sickle cell disease was one of the first genetically correlated diseases, where we knew that there was a DNA substitution leading to an amino acid substitution, leading to a structural change in the protein, leading to a disease phenotype. Okay, so it's one nucleotide substitution. Crazy, awful disease from one nucleotide substitution, which changes a glutamic acid codon to a valine codon. Okay, so glutamic acid to valine. The glutamic acid, where would you predict a glutamic acid would be on a protein?
On the outside.
On the outside, on the surface, negatively charged. So that valine substitution now puts a hydrophobic residue on the surface where there should be a hydrophilic residue. So we're substituting the negatively charged residue with a hydrophobic residue. And now what do we know about hydrophobic residues on the surface? So we sometimes call them sticky patches. So if you have two subunits and they both have hydrophobic surfaces, that's often where the two subunits come together. So now if you have a hemoglobin tetramer and a new little hydrophobic sticky patch, it's going to find the sticky patch on another hemoglobin molecule and aggregate. Okay, so you have these aggregations of hemoglobin that change the shape of the red blood cell, prevent them from going through the small blood vessels, bad stuff. Okay, so structurally and mechanistically, there are several people at Wake Forest who study hemoglobin structure, fibrin structure in blood cells. Danny Kim Shapiro, Bruce King, and Leslie Poole at the med school. Okay, so if you're interested in this, talk to them. Practice problems. That's all we're going to say about hemoglobin. Any other questions?
Do they know of any way to reverse the sickle cell disease condition at all?
So that's a good question. So what is the treatment for sickle cell disease?
Blood.
So blood transfusion, certainly. So there's no cure. Okay, so it's all disease treatment, pain management. So there are some tricks.
Because I mean, the main thing about it is it polarizes.
Mhm.
That's what causes the clot. So there's no like medication you get to prevent that.
Well,
Negative polarization.
so some of the treatments, and this is where Dr. King and Dr. King Shapiro work, is if you treat with nitric oxide, and so that's the compound that Dr. King works with, that will kind of shift the structure enough so you actually get some covalent modification of the hemoglobin that changes its shape so it's less likely to polymerize. Okay, so you're not fixing the nucleotide substitution and the amino acid substitution, but you are kind of interrupting that sequence to structure problem. Okay, so if you can prevent the aggregation, then you're going to prevent the sickling, which you're going to prevent the pain because oxygen isn't getting to tissues. Say that again?
People who are like Kalios for sickle cell, or any type of allele.
Yes. So that's the weird thing and why, what seems to be a dreadful disease persists in the population. Okay? So if you are heterozygous for the sickle cell disease, so if you're a carrier, so mosquitoes don't like your blood. Okay? So it's not that they don't like your blood, they can't reproduce in your blood cells. Okay? Because what happens when you have all these aggregations in your blood cells? The blood cells actually break. The red blood cells lyse, and then the mosquito population can't reproduce in their larval stage, and so you're essentially resistant to malaria. So there's a beneficial protective aspect, mostly if you're heterozygous, because you don't want to be homozygous and have sickle cell disease, but if you're a carrier, then you have protection against malaria. So it's a cool disease from biochemistry aspect, not from the physical toll kind of aspect. It's a yucky disease. Any other questions? We're not going to start enzymes today. I will ask you in the last few minutes. So you're welcome to ask a question if you want, but I want to ask you a question. And I don't need to record this, really. It's not a bad question. I just don't need to record it. So I am